in this unit. The first of which is the respiratory system. The second of which is the digestive system, which actually relate to one another quite a bit. You might not think about that, maybe you do, but breathing and eating are processes where we bring something into the body that our body doesn't naturally produce. In the case of the respiratory system, we're breathing in oxygen. In the case of the digestive system, we're ingesting, water, electrolytes, carbohydrates, proteins, so on and so on and so on. Okay, so we're needing to bring stuff in, and those are actually the ingredients that we need to make energy. Okay, but as a byproduct of both breathing and making energy and digesting our food, we also produce waste products that we must get out. In the case of the respiratory system, we're talking about carbon dioxide, which we've learned very recently. If you don't exhale your carbon dioxide, it can lead to acidic environments inside our body, because what it will do with water is form an acid. Whereas the digestive system, we're talking about make a poop, and we'll get into that obviously here in the next couple of class periods. So there's three major things that we can say our respiratory does to contribute to homeostasis. Number one, I would say, is the primary function, which is providing for exchange of gases, oxygen, carbon dioxide between our atmospheric air, but also our blood and our tissue cells. By exhaling that CO2, I just mentioned, that helps regulate our blood pH. And then kind of secondary functions there, containing receptors for the sense of smell, filtering inspired air, producing sounds for, talking vocal sounds, and excreting small amounts of water and heat. So we're actually going to talk a little bit about acid-base balance, regulating pH in this lecture, just very briefly. We introduced at the beginning of the semester when we talked about acids and bases and the pH scale. And I said that there are two main systems that are most responsible for regulating pH. One of them is the respiratory system. The other, we have yet to talk about it. It's not the digestive. It's the urinary. And that's what we'll get into in our last unit. Let's just start here by introducing the structures and what we would call the divisions of the respiratory system. Kind of like what we usually do. A little background on the structures and their anatomical features so that we can understand how they work. There's kind of two major ways to break up the different structures of the respiratory system. First way is to divide it structurally. The second way is to divide it functionally. Okay. If we look at the structural classification, there's two different tracts or systems, what we call the upper and the lower. The upper respiratory tract or system includes the nose, the nasal cavity, which is the inside of our nose, and then the divisions of our pharynx, which is a fancy term for our throat. Then you have the lower respiratory tract or system, which includes the larynx, which we commonly call our voice box, the trachea, which we commonly call our windpipe, and then the bronchial tree, which is a series of airways on the inside of our lungs, and then the lungs as a whole as well. Then the two divisions of the respiratory system based on their functionality include the conducting and the respiratory zones. The conducting zone is going to filter, warm, moisten, and conduct air into the lungs. Okay, hence the name conducting zone. It conducts, it moves the air into the lungs. It goes from our nose all the way down to what we call our terminal bronchioles. Then the respiratory system or zone, consists of tubes and tissues within lungs where gas exchange actually occurs. So this is where oxygen actually enters the blood. CO2 is able to leave the blood to then be exhaled out. It includes respiratory bronchioles of either ducts, of either sacs, but more importantly than any are these structures called alveoli. And we're going to learn more about them later on in the lecture. But those are these like tiny, and I'm talking microscopic sized balloon-like structures that are surrounded by our pulmonary capillaries, where the true exchange of the gases takes place. Okay. Now, those are all structures located inside your lungs, and your lungs are located in the thoracic cavity, which also consists of the heart, large blood vessels, trachea, esophagus, and thymus. Not that that's really important, but I listed it off anyway. Surrounding our lungs are two wet epithelial membranes, and they are a parietal and a visceral pleura. Okay. And these are very important because they affect breathing in and breathing out. Okay. They are pretty similar to the pericardia that we talked about the other day that surround our heart. Okay. Surrounding your heart, we learned that there's a parietal and visceral pericardium. In the case of the lungs, these are now the parietal and visceral pleura. Parietal pleura is a thin superficial membrane, okay, which lines the thoracic cavity. And then the visceral pleura, which covers the lungs. Okay. In between them is a small, and I'm just going to describe it like a potential space. We call this the intrapleural space. Okay. It is full of a fluid called pleural fluid that reduces friction between those two membranes, but also produces surface tension that causes the two membranes to stick together. Okay. And this is important because this is, as like I said, going to affect your ability to breathe in and out. Okay. Within this space is essentially a vacuum, not like the type of vacuum that you use to clean your floors, but it's a established or maintained pressure. Okay. And that's what helps keep those two membranes along with that fluid sticking to one another. I always think about like their relationship, like from a movie that I watched as a kid, Little Giants, where there's a scene where a boy, he's trying to play football, can't catch anything, so they put some glue stuff on his hands, and they send him out and they're going to throw him a pass, but right before he puts his hands together and they get stuck, and then he pulls his hands apart and they snap back together. And that's what these two membranes like to do to one another. They like to be attached. Okay, so if you pull the parietal pleura in one direction, the visceral pleura is going to go with it. And you've probably heard of somebody having a collapsed lung, right? A collapsed lung would occur when those two membranes are no longer sticking to one another. There's no longer that vacuum in the intrapleural space. Okay. Now, I'm kind of getting ahead of myself, but that's one of the things involved with breathing, which is one of the 5 processes of respiration. Respiration is a fancy term for the process of gas exchange in the body, which begins with breathing, what we would call pulmonary ventilation, if we were going to be a little more specific, where air flows into and out of the lungs. Movement of air into the lungs is called inspiration or inhalation, whereas movement of air out of the lungs is referred to as expiration or exhalation. And when we inspire, this allows O2 to enter the lungs, and when we expire, this permits CO2 to leave the lungs. And that's obviously what's shown here at the top with our figure, okay, where we're bringing in O2 and we're pushing out CO2. We'll talk about that here in just a second. Okay. Now, in this illustration, we see that term alveoli that I mentioned just a second ago. And I told you guys that's where the gas exchange occurs, where actually oxygen is able to enter the blood and CO2 is able to leave the blood. Okay. If I go back to this slide, listed down there as part of the respiratory zone are what we call alveolar ducts and alveolar sacs, along with alveoli. And I'm just going to kind of skip over respiratory bronchioles. Okay. What we see here is a cluster of alveoli, which is what an alveolar sac is. I always tell students, think of alveoli like grapes. Okay. Each individual ball-like structure is an alveolus, which is the singular form of the word alveoli. Okay. And like grapes, they don't grow in solitary situations. They grow in clusters. So a cluster of alveoli is an alveolar sac. Okay. And like the stem that holds those grapes together, that's what this is, and that's what the alveolar duct is. That's an airway that leads to all those little sac-like grapes. And that's where, and what's not shown, but pulmonary capillaries will be very prevalent surrounding each one of those for ready gas exchange. The type of gas exchange that occurs in our lungs is called pulmonary gas exchange. Also goes by the term external or pulmonary respiration. And this is where we exchange the gases between the alveolar, or between the alveoli or air sacs of our lungs and the blood in our pulmonary capillaries. In this step, pulmonary capillary blood gains O2 and loses CO2. Okay. The third component of respiration is the transport of oxygen and carbon dioxide by our blood, which relates to what we've been talking about here for the last couple of class periods, which, thinking back to the day, we talked about the blood and things like hemoglobin. We're going to talk more about that here again in this lecture. We'll get to that point today, though. Okay. So this is where we are going to be transporting oxygen to our systemic tissue cells. So from like the top of our head down to the bottom of our feet, where we're going to carry that CO2 the opposite direction to the lungs to be exhaled out. Once we get that oxygen to the tissues, we need to bring that oxygen into the tissues, which is what we do with step 4, systemic gas exchange, which goes by also internal or tissue respiration. And in doing this, the blood loses the oxygen because it goes into the cells. CO2 is removed from those cells and is pulled into the capillaries to then eventually be exhaled out. What we're not going to get into in this lecture is number 5, which is cellular respiration, where we use that oxygen, we produce CO2, we produce a little bit of water, but more importantly, and the whole reason that we do any of this is to produce ATP, produce energy. And this is going to involve processes like glycolysis that we've mentioned before, Krebs cycle, the electron transport chain, which if you've taken biology or any other form of science class, I'm sure you've been exposed to it, whether it was freshman year of high school or in another college class. That's actually going to be what we're going to talk about in the last lecture of this unit. And that's our cell metabolism lecture. And that will be what we discuss after we've talked about the respiratory and the digestive systems. Because the respiratory and the digestive systems provide us with the ingredients, the nutrients like glucose from carbohydrate-loaded foods, oxygen, which we get from breathing. So that's why we talk about those two systems and then talk about the wonderful processes of cell respiration. Okay, so let's talk pulmonary ventilation. That's our very first of the 5 steps of respiration. In other words, that's what we would commonly call our breathing processes. Consists of a repetitive cycle of inspiration and expiration. We call this constant alteration between inspiration and expiration, the respiratory cycle. We, on average, at rest, have about 12 breaths in, 12 breaths out per minute. Okay, that number can go way up or it can slow down depending on the state of the body, just kind of like what we learned with heart rate. When we exercise, your heart rate goes up, so does your respiratory rate. Okay. Now, how this all works is very similar to what we learned about when we talked about the flow of blood. And as I promised, when we talked about that, we would talk about this again. We're talking about pressure gradients. Okay. And in the case of breathing, we have several laws that we're going to see in this lecture, but the one associated with breathing is what we call Boyle's law, which states that there's an inverse relationship between the volume and the pressure inside of a closed container, i.e. our lungs and the thoracic cavity. Okay. What that means is if we have an inverse relationship, one goes up, the other one goes down. Or if one goes down, the other one goes up, right? We've seen that several times here lately, like in the case of vascular resistance and blood flow, that was an inverse relationship. So here we have kind of a easy experiment to demonstrate this relationship. We've got a closed container, okay, full of air with a piston that can change the volume inside. So in other words, can change the amount of space on the inside of the container, okay? With the piston in the up position, we say that the volume would be measured out at 1 liter. And the pressure at that point is 1 atm. That's short for atmosphere. Okay. Well, here the volume is pretty high and the pressure is pretty low. Whereas if that piston pushes downward, the volume goes down to a 1/2 liter. And in essence, that drives the pressure up because there's more stuff, or there's the same amount of stuff in a smaller space. The same thing can happen like if you think about like a juice box. okay, or a water balloon. A water balloon is a good example. Okay, if you squeeze a water balloon, what happens to the pressure inside? It goes up, right? Because there's less space for that same amount of water. Okay. Well, how does this relate to breathing? Well, we can change the dimension of our thoracic cavity where our lungs are, either increasing it or by decreasing it, which is going to affect its pressure inside. And this is what we do to inspire and expire. Okay. Inspiration is the movement of air into the lungs, occurs when atmospheric pressure, what we would say is at sea level 760 mmHg, there's that millimeters of mercury, or we abbreviate that P sub B, which is short for barometric pressure. So that's the pressure in the atmosphere that we're breathing. If that's greater than alveolar pressure, so in other words, if P sub B is higher than the pressure in our lungs, our alveoli, P sub A, we've got a pressure gradient that's going to draw the air into the lungs. Now, how do we drop pressure in the lungs? We make our closed container larger. You'll see on the next couple of slides how this all works. When we expire, we move air out of the lungs, and this occurs when the pressure inside our lungs in the alveolar spaces is now greater than the atmosphere. So P sub A has to be greater than P sub B to push the air out. So how do we change those pressures? And we have to change the dimensions. And we have to contract mainly our diaphragm as well as some other muscles, or relax it in those muscles. Okay. And we'll see here in just a second. So to kind of emphasize how it all works, alveolar lung pressure changes as a result of changes in intrapleural pressure. Remember, intrapleural pressure, P sub IP, would be the pressure in the space between those two membranes around our lungs. Okay. During the respiratory cycle, so in other words, as we breathe in and out, several mechanisms are used to change the size of the thoracic cavity, which in turn changes intrapleural pressure. So if our thoracic cavity dimensions go up, that means the space has gone up. What would happen to the pressure in the intrapleural space? We said it's an inverse relationship between pressure and volume, right? So if our volume goes up, pressure here goes down, right? Okay, if pressure in the intrapleural space goes down, so does pressure in the alveoli. Alveolar pressure drops as well. That establishes a pressure gradient that's going to result in air coming in. What if our thoracic cavity dimensions decrease? So volume goes down. What would happen to pressure here now? It would go up, right? Because it's obvious, or it's an inverse relationship. One goes down, the other one goes up. So dimensions go down. That means our volume's gone down, pressure goes up. Pressure also in the alveoli go up. Now the pressure gradient is established, that forces the air out. We exhale. So how can we change the thoracic cavity dimensions? Well, in the case of inhalation, I've already mentioned, the most important muscle is your diaphragm. Other term for inhalation is inspiration, and that's just the process of breathing in. And for this to occur, the air pressure in the lungs must decrease below pressure in the atmosphere. And this is accomplished by expanding the lungs to increase their volume. Volume goes up, pressure goes down. Most important muscle, as mentioned, is the diaphragm. This is a dome-shaped muscle that forms the floor of the thoracic cavity. And by contracting it, we can say that this is an active process, because what we've learned previously, contracting muscles is pretty complicated. This is the skeletal muscle, so it's got the actin-myosin, it's got troponin and tropomyosin, all that wonderful stuff. And it's very active. Okay, major muscle we can describe as being used when we quietly inspire. What we're all doing right now is quiet inspiration, which is just where you're taking normal breaths in and out. Okay, quiet normal breath in is quiet inspiration. The quiet normal breath out is quiet expiration. Okay. Now, the next most important muscles of quiet inspiration are your external intercostal muscles. These have the ability to pull the anterior end of each rib up and out, enlarging the thoracic cavity from front to back and side to side. Now, if everybody takes a deep breath in, take a deep breath in. You just performed what we call forced inspiration, which involves other skeletal muscles, namely the scalenes, your pectoralis minor, and your sternocleidomastoid. These are located kind of in the upper chest and even into the neck, which is where your sternocleidomastoid is. And they help increase that thoracic cavity size even more than it already was. Okay. So when the diaphragm contracts, it goes from a kind of dome shape like we show here to more of a flattened shape, okay? And in doing so, that pulls on those two membranes we just talked about, okay? And remember, those membranes stick to one another like a kid in the movie Little Giants with his hands glued together. If the parietal pleura is pulled down with the diaphragm, so is the visceral pleura. And in doing so, that pulls the lungs with them. And so the volume goes up and the pressure goes down. Look what happened to alveolar pressure and intrapleural pressure here versus now here. Doesn't have to be a drastic change in pressure, but it is enough that now pressure in the lungs is less than pressure outside the lungs. 760 is more than 758. Correct? So that's a pressure gradient that's going to draw the air from high pressure to low pressure. So I've already kind of explained it, but let me read through. As the thoracic cavity enlarges, it pulls the lungs along with it because of the cohesion between the parietal and pleural membranes. Thus, the lungs expand and the pressure in the bronchial tree decrease. The ability of your lungs and the thorax to stretch is called compliance. Compliance in the lungs is related to two principal factors: elasticity and surface tension. What you want is your lungs to have high compliance and thus can expand very easily because the elastic fibers in our lung tissue are easily stretched. And then there's also what we call surfactant, which we'll learn a little bit more about, in our alveolar fluid that reduces surface tension. Okay. Think of a rubber band. And we've talked about this before. A rubber band has the ability to stretch, but then also to recoil, right? Well, if you think about like going to Walmart and buying a bag of rubber bands, you can buy like the variety bag of rubber bands, where you've got some that are really thick and really strong, or some that are really thin and much easier to stretch. Okay? Or you think of like if you work out and you use like those TheraBands that have different colors, some are easier to stretch than others. Okay? What you don't want is your lungs and your membranes of the thorax to be like a really thick rubber band. What you want is that thin rubber band that's easy to stretch. But there are conditions, unfortunately, that can decrease compliance and thus make your lungs more like a really thick rubber band, or what is it, like the black TheraBand? That's the really hard one to stretch. Okay. Pulmonary conditions that decrease compliance include scar lung tissue, where the lungs are obviously scarred. Lung tissue becomes filled with pulmonary fluid or fluid in the lungs, pulmonary edema. There's a deficiency in surfactant, or where there's an impeded expansion of the lungs in any way, like in paralysis of the intercostal muscles. Okay. So that's decreased compliance. Okay, and the people that have decreased lung compliance, their lungs don't stretch, so they can't take like a really deep breath in. Now, what you also don't want is too much compliance. Okay, what you don't want is like what can happen to your favorite pair of sweats after a couple of years of wearing them all the time, where the waistband gets too stretched out that it no longer likes to snap back, right? It's easy to pull and stretch them out, but now they don't snap back. So there's conditions that can increase lung compliance, and this includes emphysema, which can be due to destruction of elastic fibers in their alveolar walls. Okay. And so people with emphysema don't have any problem taking a deep breath in. What's the issue with people with emphysema is blowing that air back out. and that's what exhalation is. So in the case of exhalation, it occurs when the pressure gradient resulting in inspiration are now reversed. So in other words, pressure now has to be higher in the lungs than it is in the atmosphere. 762 is higher than 760, so that forces the air out into the atmosphere. Okay. Now, unlike quiet inspiration, when you're quietly breathing out, we call this a passive process because it only involves relaxing the muscles that we just previously learned contract when we're normally inspiring quietly. So in other words, when we quietly exhale, it's basically just a passive process because it's involving the diaphragm just relaxing, going back to its original dome shape, which in doing so pushes the lungs to decrease their size, decrease volume, increases pressure, and pushes the air out. Does that make sense? So that's why we said quiet inspiration is a active process, but quiet expiration is passive. Now, what this does require, though, is a dependency on your lungs to return to their initial size, causing a decrease in the size of the thoracic cavity. And we call this ability of our lungs to return to the initial size elasticity or elastic recoil. Okay. And that's, like I said, what people that have a stretched out waistband in their pants don't have. Okay. Individuals with emphysema are like those stretched out waistbands, where they stretch really easily, but the recoil is gone. One of the telltale signs of somebody with emphysema, if you've never been around them, aside from probably having a gas mask hooked up to their nose all the time, is that their lungs, they describe it as being barrel chested. Basically, their lungs are just like overinflated all the time. They can't just puff and blow everything out. They can't even exhale completely because their lungs are overstretched. They don't snap back. They don't recoil. Okay. Now, speaking of huffing and puffing and blowing out, that's what forced expiration is. Okay. Taking a deep breath in, forced inspiration, blowing that air back out is forced expiration. That involves recruitment of the muscles of exhalation. Okay. That includes your internal intercostal muscles. So your externals help with breathing in, internals help breathing out, as well as your abdominal muscles. And they can increase pressure tremendously and establish a pressure gradient up to 20 millimeters of mercury that forces the air out pretty darn quick and pretty significantly. Does that all make sense? Now think about emphysema. Pulmonary edema can be caused by certain conditions. Lung tissue scarring. What is a common theme in a lot of those situations? In other words, what do people do that causes them to end up with an emphysema? Smoke. And now vape. Okay. A lot of lung issues are self-induced. And people know that. Unfortunately, they choose not to pay attention and do it anyway. So it's unfortunate. Now, like the flow of blood through blood vessels, the rate of airflow through airways depends on both pressure gradients and resistance. So we talked a lot about resistance in our blood vessels lecture. Systemic vascular resistance, we learned, is inversely related to the flow of blood. Well, we have airway resistance, kind of the same concept, except it's not blood that's flowing, it's air. And resistance affects both inspiration and expiration. Okay. What we want is very little resistance. Okay. And the walls of our airways, especially our bronchioles, offer some resistance to the normal flow of air into the lungs. Okay. The larger the diameter of the airway, the less resistance they have, which makes sense. They're more wide open. There's less resistance to the flow of air. Okay. As our lungs expand during inspiration, the bronchioles enlarge because their walls are pulled outward in all directions, which decreases airway resistance. And that is what happens when the diameter of the bronchioles decreases. Okay. Airway diameter is also regulated by the degree of contraction or relaxation of smooth muscles in the walls of the airways. So we talked about vasoconstriction, vasodilation, and how those can affect resistance. Now we have bronchoconstriction and bronchodilation. Bronchoconstriction, bronchodilation is essentially the same thing, except now we're not pinching or opening up blood vessels. It's airways. Okay. Bronchodilation decreases resistance because our airways are more open. And this is what we do when the sympathetic nervous system sends out its signals, which should make sense because, as we know, if we're in a fight or flight response, we're not only breathing more often, we're going to be breathing more deeply. So we want that resistance to the flow of air in and out of the lungs to be diminished, right? Whereas when the parasympathetic division is sending out its signals, we're going to trigger bronchoconstriction, which increases resistance and less air is able to flow through. But that's okay because we're in a rest and digest response. We're not needing to move tons and tons of air. Now, there are conditions that can narrow or obstruct the airways, which increases resistance so that more pressure is required to maintain the same airflow. It's kind of like what we talked about with atherosclerosis. A hallmark of asthma or chronic obstructive pulmonary disease, COPD, which includes both emphysema and bronchitis, is an increased airway resistance due to obstruction or collapse of their airways. We'll talk a little bit more about those in a little bit. Probably not today, though. Actually, you know what? I'm going to stop right here because I don't want to start this. We're not going to have enough time to get through like the next 4 slides where we talk about all this. So we'll stop right here, and then we'll start fresh with pulmonary volumes Tuesday after we get through the exam, and then run from there. We got a pretty good jump, so I'm happy with where we're at.